All right, so we're going to talk about chasing peaks today. So here's a quick little simulation showing a single character changing. We'll talk about what this means in a minute. Again, any questions, stop, interrupt me, please. All right. So let's think about how a trait can change in a single instant in time, right? So it could change randomly, right? Increase or decrease slightly due to chance, right? And or it could be changed directionally, it could be pulled towards some value, right? And so think of this as an Ornstein-Uhlenbeck process, right? So have you ever seen those tortured kids at the mall on the leash, the elastic rubber band, right? Here's this process where they're being pulled towards some value, right? And it tries to get away, and it wiggles, and it moves, pulls back, right? So it has this intrinsic wiggle property of a toddler, right? And that's what we call this sigma, right, this wiggle. And we have the strength of the rubber band pulling the kid towards you, which is this alpha, right? And then where you're being pulled towards is theta. That's the push pin or the parent, right? So that's an Ornstein-Uhlenbeck process that we've been talking about throughout the course. Okay. Any questions about that? Okay. Now we can imagine splitting that process up, right? So we can imagine having a certain set of parameters just on that little part of that edge, and then another set of parameters right here, and another set of parameters right here, and so forth, right? So you imagine this general model where we have all these parameters changing in many, many places on the tree, okay? This is probably very hard to estimate all these parameters with 3 data points or 5 data points, right? So we did have some sort of simplification, but in the ideal, and what we think reality is working like, we think it's something like that, right? And trying to approximate it with our models. And there have been various developments to do this. So here we have our sigma, alpha, and theta again. And so single rate Brownian motion has equal sigma on all branches. Alpha is 0 on all branches, no pulling at all. We've cut the kid's cord. And theta is irrelevant other than the root state. Okay? And this is tremendously useful for the various reasons we've been talking about, right? Central limit theorem, many processes adding together give you Brownian motion. Okay? So it's been used in independent contrast, initial state estimation, even more. We can try having a multiple mean Ornstein-Uhlenbeck process, right? Where now we have sigma equal for all the branches. We have alpha equal for all the branches, but it's non-zero. Okay, so you could have some strength of pull, and then you can have theta, and some of those could vary. So you could have one theta over the entire tree, right? So Amelia's PGLS had that. You could have different thetas in different groups. Why might you want that? What's the utility of this? More parameters good, right? That's fine, but why biologically do we care about these parameters? So what does theta mean in the model?
Theta is the adaptive peak.
It functions like the adaptive peak, right, which means what? Are we moving towards it, away from it, dancing around it?
Selection, or in this case alpha, will push the baby back towards the pin.
Right, so we pull it towards the pin. And so if I have multiple thetas, that means I have different pins, right? So I have, this parent over here pulling its kid, this parent over here pulling this kid. Okay, now biologically, we're thinking about a phylogeny, right? Why do we care about that? What sort of questions could you ask? All right, think about mammals, right? So think about body size evolution for mammals. What's the optimum body size for a sperm whale? Big. How about for a mouse? Well, if they were being pulled to the same optimum, and you couldn't have a mouse on land evolving to sperm whale size, right? So if they're evolving under this pulling process, they probably have different optima, right? This is the heterogeneity Joe was talking about over the whole tree, right? So probably have different processes. And so if we care about, do carnivores have different optimal body size than herbivores, we could try fitting different thetas, keeping everything else the same. Okay. Does that make sense? Okay. And so there's a bunch of papers that use that. Okay. We could have multiple rate Brownian motion, right? And there are various papers that use this. So here, we don't have any pull anymore, but we have different rates of wiggle, right? So you could have annual and perennial plants, right? Maybe perennial plants evolve more slowly because they have longer generation times. So you want to have a different rate of wiggle in those branches, okay? Maybe if we think of Brownian motion as following this moving peak, maybe peaks in some areas move faster than others. Maybe peaks in the temperate area move around more quickly than peaks in the very stable tropical areas. Okay, so we can try fitting different models there. If I'm thinking of a multiple everything model, right, where we have all three of these vary, okay, and you might say, okay, I think, and then you allow you to restrict this further. So you might say, okay, let's just assume that the wiggle rate is the same for everything, and the alpha and the theta is the same for everything, but we may have different alphas, right? Maybe I think that I'm being pulled more towards the peak here than here. Right? There's other selective constraints on them in other areas. Okay, so we can test these models. And if we can test these models, not to say this model is best, but actually get parameter estimates that make sense to us and help us understand the biology. Right? Do mice and whales have different optima? Are herbivores being pulled more strongly towards the body size than carnivores are, and so forth. So what I focus on this is this sort of general model. Now, still not completely general, right? So we typically, when we use this, assume only one set of parameters per branch, right? You could, in theory, break up a single branch into multiple ones. There's actually very little signal there to do that. Okay? But again, if you have some hypothesis about, do I think that 5 million years after the KT extinction, there was a different model just in those branches, you could take those segments of branches and just apply a different rate there rather than the rest of the tree. Okay? So you can break branches if you wanted to. Right? But there's really not a lot of power to get that, but we try. And so Jeremy Bellew, who's a postdoc, Tony, who's back there, Carl Boediger, and I developed a set of models like this. So we have different names for them. So BM1, Brownian motion, one rate parameter, the basic model. BMS, Brownian motion with different rate parameters. So you think of them as being associated with states, right? So if you had some trait of carnivory versus herbivory on the tree, you could say, do carnivores have a different rate than herbivores? So you have state dependent. Ornstein-Uhlenbeck with one parameter, one mean. Ornstein-Uhlenbeck where only the mean varies. Ornstein-Uhlenbeck where the mean and the alpha vary, mean and variance, SD, and then all three of them. Okay? And you can see the number of parameters you have. So BM1, BMS2, and so forth. Any questions about these models? No? And of course, some of them are nested within each other, some of them aren't. Right? So BM1 is nested within BMS, but BMS is not nested within OUM. Okay? So likelihood ratio test might not be appropriate, but we could do AIC or things like that. So you apply this to a data set, looking at genome size, genome size of monocots. Okay? And we have herbaceous monocots and woody monocots. Okay? And trying to estimate different optima for their genome size. Okay? And here we see the distribution of the thetas. Okay? And decimal value for some of them. So I'll show you the confidence intervals in a minute. Okay? But here's the empirical range is these rounded plots. Okay? And our actual estimate is the dotted line, and the confidence interval is the shaded part. So for optimal genome size for herbaceous plants, the green one, we see it's pretty near the peak of what we observe in them, right? There's variation around that. And for the woody plants, it's a bit lower, but we'll see in a minute, we have to do a very bad job estimating that. Okay. So what we can do is try for each of these models, we can calculate the likelihood, calculate the AIC, calculate the delta AIC, and then calculate the weight for the model. Right? So looking at this, what's the best model for our data? The last one. Right. Why do we think the last one? Right. So delta AIC is 0, and it has the biggest weight. Okay, both of those. But this one still has substantial weight. Okay. We don't talk about significance. We talk about how important different models are. And so both of these seem to be important. The others seem not to be that important. Okay. So using that best model, we took these estimates, and so here estimate for alpha, right, sigma, and theta for herbaceous plants and woody plants. Okay, so when you look at this for a minute, and then talk to your neighbor and figure out what this all means. Okay, we have a bunch of numbers up there. Why do we care about these numbers? How do woody and herbaceous plants differ? If you were to tell it in words. Talk to your neighbors. Yeah. They're not. Yeah. Okay. One more minute. Okay. So what do folks think about the biology here? What does this model tell us? What other conclusions have you drawn from this table? Come on, the people are actually talking about it. I heard you, right? They can't see your face online. They can only see me. It's okay. They see the back of your head. But all right, so, yeah.
So maybe I missed the notation thing, but
Uh-huh.
I thought that a theta was a matrix that defined the adaptive peak, right?
I think it's a vector, but yeah.
Okay, so what's
Well, it's, yeah.
so what?
Bookkeeping.
How is that like compressed to in like a scalar there? So what does a number as a theta mean?
Oh, so we're doing only a univariate case here. So you have just one trait. Genome
Okay.
size. Yeah. Good question.
uh,
Yeah. Other questions or thoughts about this? All right, so alpha. Talk about just the alpha. How do woody and herbaceous differ? So this is smaller, right? So what?
So for herbaceous plants, the pull towards the mean is really strong. So that's a tight leash.
Mm-hmm.
But for woody plants, that is a really loose leash, almost Brownian.
Right. So why is it almost Brownian? Because this alpha is very close to 0. It's so small we can't distinguish it from 0. Okay. Now is this big? It's bigger than 0. I haven't told you how big you should feel it is yet. We'll get to that. Okay. But it definitely is much more OU than for woody plants. Okay. How about theta? How do they differ? Yep, here, go back to. Yes.
So I would say it leads to the same conclusion because for the herb plants it's quite high, whereas for the other one is really low. So basically it means that the woody plants are already at the optimum because the optimum is 0.
Okay. So now use your biological intuition, right? So what do you think the genome size is for woody plants? What's the genome size for woody plants? It's probably bigger than 0, right? And yet we're saying that this is being pulled towards 0. What should you conclude about that? Is there any other data here on that row that gives you something to use?
Yeah, I mean the confidence intervals are plus or minus infinity, so
Yeah, it's pretty big
I know what
range,
that
right?
means,
So
yeah.
it looks like a low number now, but it could be a huge number, right? We have no data for that, no information about that. Why don't we have any information about that? What about the model makes us have no information?
Oh. Is it because the generation time is really long comparing to herbs?
So it could be, biologically it could be because of long generation time, something like that. So we have a lot of time for selection to be happening. What from the model tells us we're going to do a bad job estimating theta.
Since we think that alpha is really weak, the rubber band's not pulling it anywhere, so you can't figure out where it would be pulled.
Exactly. So it's basically Brownian motion, right? So we have no pull towards anything. Right? So you have this kid wandering around the store. We don't know where his parent is, right? Because there's no pull to the parent. Okay? And so, of course, the bear could be anywhere on the planet, you know, plus or minus infinity. So that's why we don't have a lot of information there about that theta. Okay? Now, this suggests that a better model than the OU MVA would be something that's OU for herbaceous and Brownian for woody, right? We haven't implemented that, but you could hack it to do it yourself, right? And see if that fits better, okay? So any questions about this example? Yeah?
Understand part of it, but let's just say that our information was different and we had a larger and a smaller theta, not one that's approaching 0.
Mm-hmm.
How would we interpret that? I mean,
Okay.
or let's just say that the alphas were not so like lopsided. What do we do with that information?
Mm-hmm. Right. So if we had a more, and hopefully, I think Steve's going to talk about this tomorrow with another empirical data set. But imagine we had, you know, realistic theta for both and realistic alpha, a similar size alpha for both, but woody was smaller. Well, it suggests that, you know, woody plants are being pulled towards smaller genome size, right, than herbaceous plants were. Which might be counterintuitive. You might think that given faster generation time and probably bigger population sizes, selection might be more efficacious in herbaceous plants, and so you have selection for more efficient genomes and the smaller genome size, right? So you say, oh, something's wrong. So maybe we're making a mistake, or maybe, you know, genome duplication happens more in woody plants, right? And so you could test that using other models. So help your biological intuition about that. Maybe herbaceous plants haven't evolved some large set of genes for making wood that constitute a large portion of the genome, which probably is not true, but that sort of idea, right? Make sense?
Kind of hand wavy, you know,
Welcome
and
to
very specific
macroevolution.
to the example. Yeah. Okay. Cool. Well,
Yeah, no, I mean, you're right. It's hand wavy. And we as biologists have to be careful to, you can only do this post hoc explanation for why this might be. And there's, you know, you have one parameter different, right? And so you can make it, there are many processes that could lead to that, right? So what's more powerful as a way of doing macroevolution is to say, I think this process happens in nature, right? Let me go out and test it. So people, for example, looking at different rates, have suggested that woody plants should evolve slower than herbaceous plants. So they went out and got a lot of data on molecular evolution rates and found that herbaceous plants evolve much, much faster than woody plants. So it suggests that they were right. Of course, there could be some other explanation too. So you make the prediction and then get the data, rather than get the data and then post hoc, in theory. Yeah. Other thoughts or questions about this? Yeah.
So this sort of pattern that we see for woody plants,
Mm-hmm.
could it result from multiple peaks? Like, for example, if we have an adaptive radiation with a lot of different niches opening, and then each taxon kind of finds its own niche, and they're scattered all over the place. So overall there is no pattern.
Exactly, because remember, if we have, you know, peaks moving a lot, then it becomes basically Brownian motion again, right? So the realm at which OU works, you can't have peaks moving too much or too little. It's either the golden zone for OU to work.
But then
So that could be explaining this. If you have this peak shifting around in different groups, then it would look like Brownian motion.
so basically each lineage could have its peak, which is stationary, but among them.
Exactly, because back in
there's many
evolutionary
different-
time, the peaks
right.
have been shifting a lot.
Okay.
Yep, perfect. Yeah. Which is again why it's hard to, so you, from that, if that were the case, you would say, yeah, there's really strong selection for genome size, but it varies a lot within woody plants, rather than here, which generates the same pattern as no selection, right? Which is why you have to be careful making conclusions.
Brian, a question here. Aren't we measuring genome size in log scale?
Yes.
So the mean really in woody will be 1 or close to 1.
Yeah, but
Right,
I forget the
so-
units. Yeah, I don't know.
It's cDNA, right? It's
Yeah, yeah.
PG. So that's one that's like 1,000 megabases or megabase pairs.
Yeah.
But if the
Still.
biology helps here, of woody herbaceous makes sense because herbaceous plants have a lot of whole genome duplication, more than woody plants. So there's obviously they obviously have larger mean from what it's been seen, and you would expect more genome downsizing from herbaceous plants too. So that will make the alpha parameter also larger. So for a person studying polyploidy whole genome duplication, these estimates make a lot of sense, right? If you see other pattern that is not this, I will wonder whether the method is correct.
Right.
No.
And actually, I'll show you the next slide how we test to see if the method's correct. One of the things, so I was in this course, I was teaching this course 2 years ago when Thomas Hansen was here, and he convinced me of the utility of having units, right? So in all biology, you have units for things, right? And in comparative methods, we say, oh, the rate of this, the body motion rate is 5. 5 what, right? The theta is 0.618. Okay, right? And so now when I do this sort of thing, I include the units, right? Because theta parameters have units of the trait you're measuring, right? Log cDNA, picograms or something like that, or megabases. And so it's really helpful to do.
A log of anything is unit free, I believe.
Hang on, Liam. Yep.
The log of the sigma square, or the sigma squared on a log scale is unitless because it's change.
Right.
So you could add a constant to everything and log transform it, and then actually that's not true.
Yeah. But you could back transform theta into units that you know and love.
But then the theta does have units.
Right.
Yeah. Even on a log scale.
Yeah.
Yeah.
Log mass.
Log of the unit that
Right.
it was originally in.
Yeah. So, including these units does help you understand the biology a lot better, which should be obvious, but, it wasn't for a while. Sorry.
So if I understood well, the best model was a model with multiple optima, sigma, and attraction, and then we have only one value. Is it the mean of these multiple values, or?
So multiple means 2.
Yeah.
We have a very primitive counting system. So here we have, 1 alpha, 2 alpha, 1 sigma squared. Now
for each,
so
okay, right.
You could extend it out. You could have 7 or 12 or something like that. And some of the methods in this area, like surface and bayou, can allow you to fit many, in theory, many regimes with many different thetas and sigmas.
Okay, but you have to do the separation by yourself at the start.
So
I have an idea that the herb and woody plants will behave differently, and okay.
So with our method and Butler and King and similar methods, you first do a priori assignment of let me paint this branch rate parameter 1, this branch rate parameter 2. Now you could use some of Thomas Hansen's stuff where you have an independent trait and a dependent trait, right? And then you can see how as the independent trait moves, the dependent trait follows it, right? So you have the theta is this thing that moves along through the time. And then also these other approaches, surface, bayou, et cetera, that will try to fit this for you. And so you'll enter the tree and the data, and it'll try fitting 1 parameter, 2 parameters, 3 parameters using various MCMC algorithms. There's been some work on how well those work, and it's not clear to me that they work well yet. So I don't use them yet. But they're appealing because you can have a hypothesis about, I think there are 4 different anomorphs, right? The ground trunk and the grass ones and et cetera. And you could test that by seeing if your mapping of thetas maps to those morphs you already identified. Right, so in theory you can do that. And people have in the past. Good. Other questions? Okay, so how do we know all this stuff works? So you can't see this very well. You can look at the slides. So we did a whole bunch of simulations, right? Every simulation we tried to estimate all the parameters and to see if we could estimate the parameters well. Okay, and for OUMV, we have a pretty good estimate of alpha, pretty good estimate of theta. When you get down to like OUMVA, the estimates for theta can get very bad, right? Huge range. Okay, and so these models can work sometimes, but not all the time. Okay. And so most people developing methods now will do a fair number of simulations that actually test to see, in the ideal case, will this work? How much power do I have? So ongoing controversies about this whole area, right? So does Brownian motion equal genetic drift? So if you read some of the literature, so Butler and King, which is a classic paper in this area, they say that why use Brownian motion is just a model that's purely neutral, right? It makes no sense to use purely neutral models, okay? However, Amelia and Hansen and Martins, yes, in the classic paper on linking micro and macroevolution, talked about a variety of microevolutionary models, and talked about the covariance structure for them. And so there are a bunch of them, they're sort of equivalent in some ways to Brownian motion, right? So drift, yep, it's Brownian motion. Cool, we knew that, right? Directional selection with random drift or with a fluctuating environment, also Brownian, right? Stabilizing selection, not Brownian, it's OU, okay? And so the problem here is that these multiple different processes give the same pattern, right? So genetic drift versus directional selection can generate the same model, okay? So it's hard to uncouple these, okay? But we can rule out, if BM is better than OU, it's not a stabilizing selection in that way, okay? So you can tell something, but it's certainly not true that, in my view, that Brownian motion is just genetic drift, okay? But again, people in the field sometimes disagree. All right, any questions about that? Yeah.
Yeah, me again. I'm thinking about whether the distinction between stabilizing and directional selection is always that clear. Because I'm just thinking, if we have stabilizing selection, so we have an optimum somewhere, but it's far away from where we are now as a population, as we go towards this optimum, it's sort of like directional selection, right?
Yep.
And on the other hand, we can have directional selection, but then eventually we get to some sort of boundary where we can't really go further, so that then it morphs into stabilizing selection where we get nearer, right? So
right. And so what's can matter also is the time scales, right? So if you think adaptive peak is over here, how many tens of millions of years will it take you to get there, right? And I'm thinking about PopGen, probably not 50 million years to get there, right? So at this time scale, maybe it's some other process, but you're right. And actually, we've noticed this in certain empirical data sets where you'll see, a theta mean that's sort of, if we have a tree like, we might see something like, these are values of 5, 6, 2, and our theta is, 4. And we have values of, 10, 12, 13, and our theta might be 600.
mm-hmm.
You say, so these are at their peak. These ain't anywhere near it, right? So what could be happening there? And so it could be a case where there's just this directional process. And if we had just, only one regime, you couldn't tell it apart from Brownian motion with no trend, right? We talked about that yesterday. But if you have some that aren't under that trend and some that are, to these models, it looks like an OU with a theta that's really way out there. And actually, after the last year I taught this, Jeremy and I got together and wrote a new OU model we call OU trendy, right? And so rather than having a theta, alpha, and sigma, we have just a general trend. So you can say, okay, this is also Brownian motion, but basically Brownian motion with a trend on some parts of the tree, but not other parts of the tree. we haven't published it yet. It's in OWI, we don't use it, we haven't tested it well yet, but it's a way of dealing with that sort of problem. Yeah. Other questions? Okay. So an idea from Hansen and colleagues looking at phylogenetic half-life. Okay, so alpha, 3.8, yay, no, right? Is it big or small? And so one way to think about it in terms we can understand is the amount of time it takes you to get halfway there to the optimum, right? So for this tree, so it's calculation log 2 divided by alpha times the tree height scaling, right? So 30 million years for the total tree height, this half-life, 8.4 million years, right? So to get to this new optimum, and this is the case of a NOVOS data set, it takes 8.4 million years to get there halfway. Okay. Now, and I don't know well estimates of selection within populations, but to me it seems really slow, right? So it could be a different process going on. Okay, so we're considering that. We'll talk about that a bit more tomorrow at the discussion. Okay, but for understanding alpha, putting it in these terms makes a lot of sense. Okay. One other note to be careful of is there are different programs that do this sort of thing, Ouch, OWI, Bayou, Geiger, and some of them scale trees differently. Okay, so some of them make the total root to tip length always 1, and some allow it to be 30 million years, and so the parameter scale might not be the same. So if you're comparing them, you have to first check that the scales are the same. Okay. And here's using Butler and King data set, and then here's the observed thetas for each regime. Okay, and then here is the observed means, right? So we see that it looks like the green ones are increasing. So all of them basically look like they're increasing towards the new mean in body size. Okay. So there are two ways to interpret this again. So Brownian motion is genetic drift, right? In which case it's nice in some ways because you have these interpretable parameters versus BM describes some sort of process, can be many different processes, right? So the parameters might be pop gen or might be something about how selection moves, right? This is process-based and this is phenomenological. So you prefer to be in this area, I would argue we're in this area. Okay. Another issue to be careful of is Brownian motion a restriction of OU, right? So you'd imagine if I have an Ornstein-Uhlenbeck process and alpha goes to 0, what should happen to the likelihood? All right, so Brownian motion, alpha approaches 0. Oh, sorry, Ornstein-Uhlenbeck, alpha approaches 0. What should that be? Brownian motion, right. And so in some of the programs, that happens, right, where as you get a very, very low alpha, the likelihoods converge. But in some, they don't. So in OUCH, it goes to a different number because of how they handle the root state. And so that's something to be careful of when running these as well. Okay. Bigger issues are identifiability, right? So I showed you this plot, a similar plot yesterday, right? We're talking about Brownian motion with the trend, and we can't separately identify the starting state and the trend bias, right? Here's something scarier, where we have the ancestral state and the selection optimum, theta, basically, what they call it mu, right? Where we have this ridge, and so they might not be separately estimable, okay? So it's an important paper by Ho and Ane, came out last year. And certain kinds of trees, like this kind of tree, can be non-identifiable in terms of the parameters, right? Which means that if you have a tree where you have like one clade of this, one clade of that, and the rest sort of scatter rest from a connected component, then you might not be able to estimate these thetas well, or at all, in fact. Whereas if you have things that are more interdigitated, then it might be possible. So you need homoplasy to do this. So hooray for homoplasy in this mapping trait. Okay. So this is also something to be worried about with your data set. Right, so if you're comparing, this clade versus its sister, you might not have any information about what the thetas are between them. Okay.
In that case, literally the case on the left that it is unidentifiable, or is it just not have much power?
I think it's formally not identifiable. Yeah. So I mean, the distinction is, you need a ton of data to get a good estimate. So that's sort of soft sense identifiable, unidentifiable. Or versus no matter how much data you throw at it, you'll never be able to identify it. And I think it's the latter case, but I should check that. Anyone else know? Yeah.